So again, in animal viruses, you have attachment, penetration, uncoating, biosynthesis, and assembly. Now, an animal virus is going to exit from the host cell through the process of lysis or the process of budding. With lysis, you essentially have the virus is going to disrupt the cell, the animal cell that it's been infecting. Through budding, you simply have a release of the virus as it's fusing or being pushed out of the animal host cell that it's been infecting. So when a virus is able to escape a host cell through the process of budding, then typically it's going to become what's known as an envelope virus. And the reason for that is because the virus will pick up some of the membrane that it's fusing out of the animal host cell, and so that envelope becomes a part of the virus itself. And here's kind of how another outline again of the herpes virus. This is a herpes simplex virus that's growing on a cell line called HeLa cells. And you can see in this image you have the process of absorption, which is the same as attachment, and then you have penetration taking place again, penetration again, so you can see the virus as it's going down into the cell. And then you have the process of uncoating where a coat is lost, and as the uncoating process takes place, you can see that the envelope surrounding the herpes simplex virus is going to be lost. So that's what these images here are outlining for you. This is a mechanism by which some viruses use to integrate themselves into a host cell, because remember, viruses are parasitic. But viruses, when they integrate themselves in a host cell, they sometimes can just lay latent and really not impact the cell at all. So there are a number of viruses that are considered to be latent virus infections. And a virus is going to be considered latent if the virus is able to hide itself and evade the immune system of a host. So it does this by simply remaining dormant once it's entered a host cell. And some common examples of this are some viruses that are found in the herpes family. So herpes virus, some of these can lay dormant, which is why after you've been infected with something, you may not see a reaction until later on in life. Something in your body gets triggered, and then it turns on or activates the genes that are encoded for that herpes virus. And so herpes viruses, whether acquired in the form of cold sores, genital herpes, or chickenpox or shingles, they will not get out of the host immune system at all, so they will never completely go away. And so you may have kind of an outbreak or an infection later on in life. So one example is that of chickenpox, which a patient may have earlier in life, and then as years go by and your chickenpox has resolved, you may later develop the disease known as shingles. So both of those we know correlate to being infected with the same virus. In our examples, the shingles more so than the chickenpox is an example of an infection acquired from a virus laying dormant for a number of years. So how do we treat different types of viruses? Well, we rely on antiviral agents to do this. Remember, viruses are not bacteria, so we just can't target things like the peptidoglycan structure, which was what we would use if we were trying to kill a bacteria cell with an antibiotic. We also would have, we also don't have, a nucleus to go after. There also aren't any ribosomes for us to target. So with antiviral agents, we can't rely on antibiotics to defeat viruses if they're invasive in the body. So we use antiviral agents, and these are drugs that are used to treat viral infections. And the way they do this is that they play a role in interfering with virus-specific enzymes and any process involved in the process of viral replication or production. So messing up these replication processes will prevent virus DNA and RNA and viral proteins from being created. So those are the kind of targets that our drugs are built to combat, to prevent viruses from doing a lot of damage to the body. And so we use mostly vaccines for this. Our kind of societal tactic is to vaccinate the public, generate herd immunity, so that way once your body has encountered a virus, it will know how to immediately destroy that virus or any cells infected by that virus in a very quick response time. So that's what vaccines are all about. And the way that we do this is we can take part of a virus and we can weaken that virus so that way it's not as pathogenic as it was before, or virulent as it was before. So we attenuate it or weaken it, and then we take some of that and we inoculate a patient with it, and then that patient will start to develop antivirals against that particular virus. So that's the overall basis principle of how we use drugs to combat viruses. Now there are some viruses that I said before related to having cancer or inducing cancer in patients. These are called oncoviruses. So another term for them is called oncogenic viruses, but oncoviruses is what I'll use throughout this presentation. And oncoviruses cause cancer in patients. Examples are the Epstein-Barr virus, the human papillomavirus, and then the HTLV1 virus, which is a virus that's linked to leukemia. The human immunodeficiency virus, which is also known as HIV. This is also another retrovirus because it develops acquired immune deficiencies in patients, which we refer to as AIDS. These viruses they enable cancer to be developed inside of patients because for some reason these viruses, they hijack the genetic material of normal cells and they cause those cells to massively proliferate, which is what cancer is. So nobody knows why or how this happens, but essentially what is thought to be is that you just have an acceleration in the proliferation of cells. So something's going on in enhancing the process of mitosis, which gives rise to the cancerous cells. And then what you see along the lines is that other cells start to be infected or impacted. And so you have this essential infection of cells, which is which leads to massive proliferation, which develops cancer. And when you start targeting very specific cells, such as the HIV virus, you have specialized cells known as CD4 plus cells, which are cells instrumental to the immune system. And once those cells become immunocompromised, then that weakens a patient's immune system. So with something like HIV, because CD4 plus cells are targeted, that means that a patient's body is not well equipped to fight other infectious agents, and so you actually have fatalities not due to the HIV virus, but actually due to other viruses and other pathogenic bacteria that are able to take advantage of a system that has been severely weakened, which is why HIV is called the human immunodeficiency virus. And here's an image of an HIV virion. Again, you have your genetic material that's situated beneath this capsid layer, which are all these proteins, that protein coat that's around your genetic material. And then all of that is surrounded by membrane. This is an envelope virus. And then you see all these GPs and P18s and P24s. These are just proteins, And then these are glycoproteins, which remember is used for attachment purposes. There's also an enzyme that's known as reverse transcriptase, which is very interesting because reverse transcriptase will allow this RNA to be transcribed into DNA, which, if you remember, central dogma theory is counter to the central dogma theory, which states that DNA is converted to RNA. Well, HIV contains an enzyme known as reverse transcriptase, and so this RNA will be converted to DNA. And then after converting it to DNA inside the host cell will then convert that viral DNA into viral messenger RNA and then into viral proteins. So that's how HIV is able to do its scary work and make individuals immunocompromised that are infected with it. Okay. So let's talk a little bit about viroids and prions. These are very much more simpler infectious particles than your traditional viruses that we've talked about. A viroid is simply a naked fragment of a single-stranded RNA. And then this single fragment really can start to mess up certain processes inside of a cell. And in particular, viroids can start to impact the metabolism of plant cells. Viroids are pretty much transmitted between different plants the same way that viruses typically are transmitted. Through the process of touching, through the processes of being carried, or just simply exposure. And some examples of illnesses brought in plants that are caused by viroids include the potato spindle tuber as well as Citrus exocortis, which are some interesting effects that you'll see on plants when inundated or when susceptible to these diseases. With the potato spindle tuber, what you get is simply very small potatoes that start to develop, and they are typically spindle-in shaped, which is what the name implies. They can also reflect cracks in them. With Citrus exocortis, you simply have a very small growth of citrus trees. So the height of the citrus trees is typically stunted due to this virus. And then there's a number of other diseases that can exist in things like tomato plants or chrysanthemums. But for the most part, viroids are typically associated with plants and are not going to be very common in terms of eliciting animal diseases. So the prions are our next kind of subgroup of acellular microbes, and as I've said before, these are infectious agents that are proteins, and they will cause very severe diseases, but they do this because they cause other normal proteins in a system to conform to irregular protein structure patterns. So the thing about proteins is that their structure is indicative of what their function is going to be, and if you alter or denature that structure, then the protein is no longer functional. So a prion is a small infectious protein that can influence other proteins to become small, infectious, and basically denature themselves. So they're kind of like the, just the bad influencers. So things that are caused by prions include scrapie, bovine spongiform encephalopathy, which is also known as mad cow disease, and then Creutzfeldt-Jakob disease. So these diseases are very much rooted in the nervous system, and so these prions really affect your neurons from functioning properly and are unable to generate different neurological diseases in animals. Prions are very difficult for us to understand, and they're very hard to get rid of. They're very resistant to different disinfectants, so it's not like bacteria where you can add bleach and just kill everything. Disinfectants aren't going to do anything to the prions. The prions can still be infectious. And we really don't know how prions cause disease, other than we know that they may play a role in altering the shape of your traditional